Okay, I think we should start. We have a very full program this afternoon. And the first speaker is Cori Bargmann, and I'm just going to tell you a couple of things. Last night I watched the late evening news, and there was a lot of coverage of the big event, the Breakthrough Prize at the Ames Research Lab. And there were all these celebrities. And you saw Conan O'Brien and Kevin Spacey. And you kept looking for a scientist. There was one scientist, Cori. She was sufficiently glamorous. Now, I have a bit of a beef with Cori, and I've been planning to talk to her about that for a long time. She was at UCSF, and we were really upset when she left. And she could have done the reverse. You know, we could have Richard Axel and Cori here. But instead, she went the other way. And since I love both of them, it's a really great pleasure to introduce Cori and congratulate her.
Thank you, Stan. I think it's especially sweet for me to be here giving this talk because of the many years I spent at UCSF really growing as a scientist and a neuroscientist. And it's also especially sweet to be receiving the Breakthrough Prize together with Bob Weinberg, my PhD advisor, which really adds a little bit extra to the moment. Humans have been studying the behaviors of other humans and animals as long as we have been interacting with the world. But the first scientific studies of behavior really began about 100 years ago with the roles of the neuroethologists, who in a very thoughtful and analytical way examined the behaviors of different animals. And what they observed when looking at courtship behaviors, or the behaviors of rearing young, or the complex foraging behaviors of insects, or aggressive behaviors, was that behaviors had patterns, that they followed certain kinds of patterns that were recognizable in all individuals of a species. And the way we understand that now is that it means that behaviors must have some sort of a genetic foundation. There must be some sort of genetic encoding that enables every bee to know what it should do within the hive, or that enables this animal to recognize a mating partner, or this animal to recognize its mother. Now, how something as complex and various as behavior is generated by something like a fixed genetic code is a fundamental mystery, but posed really in this level almost 100 years ago. Now, the second thing that the neuroethologists noted that I think is also a very interesting feature is that there's a difference between what an ethologist or a psychologist would call a behavior and actions. That behaviors, like mating behaviors, are composed of many different actions. And the only way you can tell the difference between a mating behavior, say, and an aggressive behavior is by the structure of actions over time. So these things develop over a certain amount of time, and this larger structure of the behavior somehow encompasses something that can develop in a very variable pattern from instant to instant, and yet over time develops this overall structure. So how do these two ideas fit together? It's these two ideas of innate behaviors and complex long-term structures of behaviors that I'd like to talk about today. And to give a sense of how those connect together, I'd like to talk about studying behaviors in simpler systems. Like many neuroscientists, perhaps most, I'm primarily interested in the human brain and behavior, but I'm intimidated by the incredible complexity of the human brain, by the billions of neurons and trillions of synapses that are in the brain of each person here. There are more neurons in your brain than there are human beings on the Earth. There are more synapses in your brain than there are stars in the galaxy. This is not something to attempt to understand in its entirety just at this moment. But this incredible complexity, and superimposed on that, the complexity that is added by experience and by culture and by learning, is actually composed of a remarkably simple number of biological components. Only about 25,000 genes make up the entire human brain. And these may be the simplest way to understand what it is composed of and how it functions. Now, among these, I will make a point that there are about 100 that encode small molecules called neuropeptides used for neural communication. It's those that I'll be talking about today. So how can we break this problem down? Well, I studied the brains of the tiny nematode worm Cenorhabditis elegans. It's 1 millimeter long, the size of a comma on a printed page. It lives in gardens. It eats bacteria. But it's an animal. It learns, it moves through its environment, it interacts with other members of its species, it detects all kinds of different stimuli. And so this complete system provides a very different level of complexity in terms of understanding the relationship between an animal, a behavior, and its brain. And that's particularly notable because its nervous system is composed of only 302 neurons in one sex, the hermaphrodite, which is basically a modified female, or 389 in the male, compared to the billions of neurons in your own brain. And yet, at the same time, this animal has almost all of the same genetic components as are found in the human brain, and perhaps soberingly, almost all of the genes that we think are really interesting for human functions, evolved functions, the genes, for example, that Eric Lander mentioned that are implicated now in schizophrenia, go all the way back to the nervous system of this tiny animal. So if we want to decode the relationship between genes and the nervous system and behavior, we have here genes and behavior and a very simple nervous system in which to interpret it. A nervous system where we have identified cells, where we can recognize every cell in the animal. Their connections are known because of serial section electron micrographs that have solved the complete structure of that nervous system, and where we have powerful genetic tools to make the relationships between genes and behavior clearer. So what's an example of trying to decipher this process? So I'll start with a well-known example. The first neuropeptides ever to be identified and synthesized in mammals, oxytocin and vasopressin. These are small neuropeptides. They're 9 amino acids long, and they regulate mammalian reproductive behavior. So when a female mammal gives birth, her system is flooded with oxytocin that is released from her hypothalamus into her bloodstream. And this causes the uterus to contract to induce labor. It causes the milk letdown response to allow her to nurse her young. And it also stimulates changes in her brain that stimulate the formation of the mother-pup bond that will then tell her that this is the most important object in the universe and she has to look after it. So these molecules are not only present in mothers, but they're present in all animals at all stages of development. And they're released under the brain under somewhat poorly understood circumstances. Studies in a variety of different animals have shown that they are involved not just in maternal behaviors, but in other kinds of social behaviors, such as, for example, social recognition in rodents. And notably in the rodents called voles, which come in different species, differences in pair bonding versus non-pair bonded behavior have been ascribed to variation that occurs between species in the receptors for oxytocin and for this closely related neuropeptide vasopressin, which also plays a role in reproductive behavior. So a couple of years ago, Jennifer Garrison and Evan McCosco, a postdoc and a student in my lab, noticed that the genome of the worm C. elegans had a molecule that was closely related to oxytocin, as well as 2 molecules related to oxytocin receptors. These molecules actually preserved their relationship to each other. This peptide activated these receptors. So biochemically, these genes exist and they're present. But really, the presence of the gene is not what's interesting. The question is what those genes are doing in the nervous system. And what Jennifer found was that nematocin, which is the worm name for worm oxytocin, nematode oxytocin, and its receptors are required for effective reproductive behaviors. They're required for this little slender male here to mate successfully with a hermaphrodite partner. Nematocin is made by neurons in its specialized mating structures and received by many other neurons in the specialized mating structures. And mating success is reduced by about 2/3 in animals that lack this signaling system. So there's a general role in reproduction, but what we can do in a worm that we can't do in a mammal as easily is to really understand in detail both how that molecule is acting at each step of the behavior and how that is being translated through the nervous system. So the first question is how this behavior takes place. So in worms, as in other animals, reproduction involves something like a courtship, a series of different steps that develop over several minutes in a certain order with a certain set of responses that are required to proceed to the next step. And when Jennifer looked closely at these different stages of mating, she found that every individual action of mating was present in animals that lacked this conserved neuropeptide in its receptor. Every turn could be made, every particular kind of action could occur, but the sequence of these actions and the transitions between them were disrupted. This is truly a global regulator of mating that keeps the sequence going in the right order and at the right strength and allows it to proceed properly. Now, this global role can be interpreted and understood in the context of the neurons that give rise to mating behavior, which have been characterized in C. elegans by a variety of other labs. So within the C. elegans nervous system of the male, there are many neurons that detect sensory cues that are important for mating, like pheromones and different kinds of touch and posture. These neurons form fast chemical connections with other neurons that generate motor behaviors, actions like contact, turn, and searching. Nematocin is not required for these systems to function. However, it appears to be induced by some of the same signals and that then acts on these circuits to change the strength of the different elements of these circuits to a behavior appropriate for mating. Nematocin is essentially providing a context cue that is rewiring the fast circuits to make certain things be interpreted in different ways. So, for example, that touch to the body, which under circumstances could signal an obstacle or even a predator that should be escaped, is now interpreted in the context of a mating behavior as a mate that should be approached. And this kind of rewiring of coordinating neurons over time, we think, is the fundamental activity of neuropeptides. So the conclusion of this part of the talk is twofold. The first point is that neuropeptides are not only molecularly conserved across a huge distance of evolution, and in fact there are at least 8 families of neuropeptides that reach all the way back through all of the different vertebrate and invertebrate animals, but that they may be functionally conserved as well. That previous work in mammals had shown that oxytocin and vasopressin were involved in mammalian reproductive behavior. Our work shows that they're involved in nematode reproductive behavior, and work from Bill Kristen's lab at UCSD in parallel has shown that they're essential for reproductive behavior in this third branch of animals, the leeches and mollusks. And what that indicates is that very likely these molecules got locked into reproductive behavior incredibly early in the evolution of animals. And that while the actions associated with reproductive behavior have changed over evolutionary time and become much more complex in animals with more complex brains, that there is a set of molecules that trigger this kind of mating behavior and help to regulate the context that reaches back into evolutionary time. As Keith Yamamoto would say, reproduction was not invented last Saturday night over a beer. These behaviors have really been important for as long as there have been animals with 2 sexes. And so the idea that these would have some fundamental component in the nervous system is not unexpected. So, how do we understand the role of these kinds of molecules in detail in a nervous system? This is a map of the C. elegans nervous system. It's a structural map, a wiring diagram, at really a level of resolution that we can only dream about for the nervous systems of other animals. Each of the 302 neurons here in a hermaphrodite has a name. The connections between them, these gray lines, are known. Their functions are known. The red neurons, for example, are sensory neurons. The blue neurons are various kinds of collecting neurons and integrating neurons. The green neurons are motor neurons. Through work in many labs following from this heroic reconstruction of the wiring diagram from John White and his colleagues, in the field of C. elegans, we know a great deal about the role of many of these neurons in individual actions. We can point to a neuron and say, this neuron is required to sense a particular odor, or this neuron is a backward command neuron that drives reversals. Through many genetic and molecular studies over many years, we know many of the rules that give these neurons their unique characteristics, and we know the rules that allow them to connect to each other. For example, work in C. elegans, and I should say also in many other animals, has led to an elucidation of the molecules required for the unique properties of sensory neurons, in part work in my lab. And work in C. elegans and many other animals has led to the identification of the rules for axon guidance and synaptic connections between those neurons, again, including some work in my lab. But I don't really want to talk about this general map. I actually want to point out now that this general map actually falls short, which is that even if you understand actions, somehow you aren't really understanding how these circuits change and how they give rise to behavior. What is it? How is it the behaviors are really manifested? And so I want to turn now for the rest of my talk to really thinking about how these kinds of small molecules, like oxytocin, are involved in regulating the functions of those fixed anatomical circuits that are composed of uniquely identifiable and reproducible neurons. So neurons have three major ways of communicating with each other. The best studied is the fast chemical transmission that occurs at synapses, and it's this form of transmission that was just recognized by the Nobel Prize in Medicine this year. This is accurate, it's local, it can occur in milliseconds, and it involves just a few conserved neurotransmitters like glutamate and GABA and acetylcholine. This is what allows neurons to fire action potentials, so it's the study of electrophysiologists. It's associated with specialized structures you can see in the EM, and so it's been the traditional study of neuroanatomy as well. That's what that worm wiring diagram is. About 90% of the synapses are in this form. Another 10% of the synapses are electrical synapses, a different form of communication between neurons. But superimposed on those is a third form of transmission, the neuropeptides and neuromodulators. Neuromodulators are molecules like serotonin and dopamine, for example. These molecules aren't released at anatomically defined synapses. They're released extrasynaptically. And they don't tend to regulate ion channels most of the time. Instead, they activate G-protein coupled receptors that couple to many different cellular signaling processes. So they're not respected very much by neuroanatomists, and they're not respected very much by neurophysiologists. But nonetheless, there are many reasons to believe that these are important molecules in the regulation of behavior. And those reasons come from genetics, and they come from pharmacology. So, for example, the disease narcolepsy cataplexy is associated with the loss of a specific neuropeptide that's made by just 2,000 neurons in the brain. The regulation of sleep and waking states is simply not normal if those neurons are not present. Hunger and pain, emotional bonding, and mood and addiction are other examples of behavior that are very importantly regulated by these neuropeptides and neuromodulators. The psychiatric drugs that are prescribed to psychiatric patients largely act through these systems. Opioids for pain control, the drugs of abuse like cocaine are all acting on these systems. So these seem to be really important for regulating long-term structures of behavior. In mammals, these things are dispersed throughout the brain, although many of them are made in the deep structures of the brain. Some are made within the cortex, many are released in the cortex. And there are many questions to ask about these, how they really work once you know what the molecules are. Biochemically, we know that they activate G-protein coupled receptors, but how do they act at the level of circuits and systems? How do they really act at the level of behavior? So we'd like to understand things like, what is the structure of neuromodulatory circuits? At what distance do they act and at what time scales? And in terms of things like wiring diagrams of the brain, they're in terms of their general not getting any respect, they haven't gotten a lot of respect there either. The sort of two dominant models for how they functioned in the past were, first of all, that they were co-transmitters was one name for them, that they were sort of mother's little helper that could assist the fast transmitter maybe by giving it a little boost, or the somewhat condescending name of sprinkler systems, suggesting that the brain was just getting spritzed with a variety of these different transmitters, and at some level those regulated these important arousal states and behavioral states. So what I'd like to tell you today is that we think neither of these recognizes completely the complexity and the interesting features of these neuromodulatory circuits. So how do we study them? We've been studying a particular behavioral kind of behavior that's controlled by neuromodulation, which is general arousal states or activity states. So the extreme of arousal states and activity states is sleep and waking, but all animals and humans tend to punctuate their behavior into relatively long-lasting behavioral states of being more active and being more quiescent. And in the foraging literature, in the behavior literature, this is sometimes called exploration versus exploitation. You see it in wild animals foraging for food. You see it in lab animals. And this is a movie of worms showing this behavior, which was actually first described by Steve McIntyre's lab here at the Gallows Center at UCSF. And what you'll see are worms going through a behavior where they sit quietly in one area and feed for a while, and then they tear around and move to another area. So here's an animal doing something called dwelling. It stays in a small area, doesn't move around a lot. Oh, a little bit of a movement. Oh, that's called roaming, okay? It just took off, went way over there. Roam some more, dwell a bit, roam, dwell. And This movie is sped up about 100 times, which makes swarm movies much more exciting. These behaviors are apparently spontaneous. They seem to be structured in events that last for a few minutes. In each case, the animal is awake, it's moving, it's feeding, it's laying eggs, but there's an obvious structure to it. And if you track the animal's behavior, and the rest of the work I'm going to talk about is all the work of Steve Flavell in the lab, who did this, you can see that you can divide the animal into these different behavioral states, the sort of high speed state and the low speed turn-a-lot state, just by tracking these two parameters. So this is the dwelling state where the animal stays in one area, and this is the roaming state where it explores. And both of these are quite long-lasting. They're 8 or 9 minutes long, which is really very different from the time scale of fast transmission, which occurs in seconds or milliseconds. So these two states, Steve found, were controlled by two of these neuromodulators in reciprocal ways. One of these neuromodulators is serotonin, a well-known neurotransmitter, the target of antidepressant drugs used in humans. And what happens in animals that lack serotonin is that they spend a lot of time roaming and very little time dwelling. And conversely, animals lacking a particular neuropeptide spend an enormous amount of time dwelling, but hardly ever roam. Now, the interesting thing about both of these is that these animals, when they are in a roaming state, perform the right actions. And when they are in a dwelling state, perform the right actions. It's the overall behavior over time. It's the duration of those behaviors that differs. It's the binding of information over minutes that seems to be the role of these neuromodulators. Now, what we can do in C. elegans is that we can take systems like this, which are found throughout brains, and really figure out how they work at a circuit level. Because, for example, there are exactly 11 neurons in C. elegans that make serotonin. And so we can ask exactly which of these neurons are important. Is it all of them? Is it one of them? Is it the overall level? Is it just some subset? And similarly, we can do the same thing for the serotonin receptor, or for the PDF receptor, or for PDF itself, using sort of classical genetic methods of conditional knockouts where we can eliminate the functions of each of these genes in different subsets of the neurons that normally make them. And from that, we were able to map out a map of the way that these circuits work. And the answer is that these circuits are neither broad sprinkler systems, nor are they point-to-point co-transmitters. They have their own separate logic. For each of the modulators, there are a couple of different neurons that make it, not all of them and not one of them. For each of the receptors, there are a couple neurons that receive it, not one of them and not all of them. And these are somewhat different from each other for these two reciprocal behaviors, although there's a little bit of overlap. And the thing that's interesting from us, from the point of view of the wiring diagram, is that these are not co-transmitters, because the neurons that are connected in this circuit here are not the neurons that are connected by anatomical synapses. It's a completely separate logic. The neuromodulators are functioning on different neurons from the neurons that are the targets of the classical transmitters made by the same cells. So how can we ask, so we now have a circuit logic. Now, how does this circuit really work? When and where do these molecules act, and how do they instruct the behavior? Many tools from genetics have come into neuroscience and created powerful ways of addressing the functions of nervous systems in real time. One of those important tools is the ability to use genetically encoded indicators of neural activity to track the activity of neurons in live animals as they move freely through their environment. And we can do exactly that with the neurons that are important in roaming and dwelling behavior. We can look specifically at the neuron that makes serotonin, for example, that's most important in controlling dwelling behavior, and ask, when is that neuron active? How long is it active? How does that correlate with what the animal is doing? And we can do that allowing animals to move freely through their environment while we track the different behavior patterns and the activity of the neuron in the course of that experiment. So using these methods, we found that the important serotonin-producing neurons have an interesting behavior pattern. They have patterns of activity that last for a minute or two, much longer than most neurons would last. And these long patterns of activity correlate beautifully with the dwelling behavior. They're time-locked to what the animal is doing. So speed, for example, is reduced in a dwelling-like behavior when the activity of NSM is high. And the reciprocal result holds for roaming. So these molecules are not just sort of generally allowing this to happen, they're time-locked to these behaviors. And the relevant neurons have the same slow activity or a related slow activity to the minute-long time scale of the activity. We can also ask how strong this correlation really is and whether it's sufficient for activity using other tools recently developed in neuroscience by a variety of different groups, notably Carl Diceroth's lab at Stanford, to activate and inhibit subsets of neurons of interest using light. So for example, we can activate the neurons that make serotonin or silence them. We can activate their targets or silence them. And we can ask how imitating the normal pattern of activity leads to effects on behavior. And what these experiments show is that serotonin is not just correlated with behavior, it's actually sufficient to induce and prolong behavioral states. That stimulating serotonin-producing neurons with light converts animals that are roaming into animals that are dwelling almost instantaneously. And that this behavior actually outlasts the stimulus, that we're seeing sort of a long-lasting representation of the behavior that matches the natural long-lasting representation. So what do we learn from these studies? How do we think about them? We think that when we go back now to the worm wiring diagram and we look at its patterns of activity, we can take the information we knew about the chemical synapses, shown here in black lines, and understand them as we did before in terms of fast locomotor actions. So these are the pathways that allow the animal to carry out its different tasks. But that overlapping with that and orthogonal to that are different kinds of modulatory circuits that give rise to long-lasting behavioral states. Now those neurons are shown here in blue. And the point I want to make here is that if we had been picking neurons at random from the wiring diagram, we would have had a hard time picking a more dispersed set of neurons from the set of connections than the ones that we find to be functionally critically linked together in regulating these long-lasting behavioral states. The logic of these modulatory circuits is separate from the logic of the fast circuits. We're going to have to study that independently. We're going to have to study it independently at the level of the neurons that produce it, and we also think independently at the level of the time scales on which those behaviors are manifested, on the longer time scales of G-protein coupled receptors and of biochemistry, which are superimposed on the shorter time scales of action. And returning to the theme of psychiatric disorders and the function of the brain, I think when we look at patients who study from many severe psychiatric disorders, there's no reason to think that there's that much wrong with their fast synaptic connections. They can act, they can think, they can throw a baseball. Those are the actions that occur over seconds or milliseconds. Really, some of these longer lasting states, the binding of information together, behaving appropriately in different contexts, may be the place to look for some of the important changes. And finally, I just want to point out that this is going to be an important feature of all nervous systems, not just those of the worm. That there are many circuits that are rewired with neuromodulators, that the strength of circuits is changed. They're important in feeding behaviors, starting from invertebrates like crabs and lobsters and going all the way up to our own feeding behaviors and obesity. They're important in sensory circuits. The best understood circuit in the vertebrate brain is the circuit in the retina that allows you to sense light. And this circuit switches between dim and bright light function, in part through the action of the neuromodulator dopamine. And they're very important in important motor circuits and in disease. And I just like to highlight this year's Breakthrough Prize winner, Malin DeLong, whose analysis of the circuits of the basal ganglia of mammals, which include important roles of dopamine, have really been critical not only for understanding the disease of Parkinson's disease, but in developing the first new therapeutic mechanism for Parkinson's disease since L-DOPA, deep brain stimulation. So by understanding these kinds of mechanisms, perhaps we can devise smarter neurological interventions as well as smarter psychiatric interventions in the pathways that allow our brains to function. I've been fortunate to have a wonderful thesis advisor, Bob Weinberg, and postdoc advisor, Bob Horvitz. I learned an immense amount at UCSF here. I particularly want to mention Ira Herskowitz, that I felt like really helped me to understand how genetics could be used to study any problem, including neuroscience. And my own students and postdocs are a constant source of joy to me. The work that I talked to you about here is Steve Flavel's, but I could have talked about many other things with equal enthusiasm. Thank you.
Questions.
Anyone want to ask a question?
Questions for the speaker?
I'm getting my instructions here. I'm supposed to ask if there are questions.
So that was wonderful. Thinking about the human brain, as you've been doing some thinking about the human brain, if neuroanatomy isn't going to be enough to tell us about the connections that matter, how are we in humans going to map these extra fast synaptic connections?
So I think understanding the maps of the human brain are, like the genome, there is not going to be one map of the human brain. There are going to be many maps of the human brains. There are going to be the different maps that exist in different people. There are going to be the different maps that exist under different neuromodulatory states where different connections are stronger and weaker. And I think that the trick is just to realize that there's a structural map. There's a map of activity in the sense that classical electrophysiologists view activity. There's the map of large brain region activity that people see by fMRI. And there are going to be maps of neurochemical activity. Once you realize it's a problem, you can figure out how to solve it. And in fact, there are experiments that go on now with methods like PET that enable people to look not as well as they should at some of these modulatory inputs. But, yeah.
Keith?
What does it mean to you, Cori, that the number of neuropeptides in the human brain and the C. elegans nervous system are the same from 100?
It's the same problem as we reach with genetics every time we look at the worm. The lack of innovation at the genetic level, of the level of certainly protein coding genes, compared to the incredible innovation at the anatomical level in the vertebrate brain and the vertebrate body is just astonishing. I mean, the only real things we can point to as vertebrate innovations are probably in the immune system and outrunning pathogens. So it shows you, I think, that the vocabulary of biology is incredibly powerful and that it can be used to construct sentences of many different sorts. The worm is a limerick and the human brain is the works of Shakespeare, but the word means the same thing in either case.
Okay.
Bruce Ames. Both oxytocin and serotonin are controlled by vitamin D hormone. We're just working. Rhonda Patrick in my lab is just showing all of this. So it'd be interesting to see whether the genes there also are controlled by vitamin D hormone.
That is really interesting. I don't know. That hasn't been looked at.
Okay, one more. Keith?
I feel like I'm taking a qualifying exam. UCSF colleagues here.
Cori, your movies showed that, or at least implied, that roaming and dwelling are reciprocal behaviors, as if there's a switch that is embedded in the circuitry. Your circuits showed that they're overlapping. They're distinct, but they are overlapping. So there's common components there. Have you thought about how you construct a switch from components that have overlapping elements?
Yes, and the logic of the circuit switch between roaming and dwelling is just the sort of bistable switch that you might see in gene expression. I always like to mention transcription when Keith asks a question. And that works well in qualifying exams, too, in case there's any students in the room. And in fact, for example, probably one of the most interesting effects, if you eliminate both the system that drives long dwelling and that drives long roaming, the combination can both roam and dwell, but it's very unstable. It switches back and forth incredibly rapidly between these two states, which is exactly what happens. It's sort of an invasion or lack of stability of states is, for example, exactly the characteristic of narcolepsy when you disrupt peptide signaling in the human brain.
Okay, let's stop. We're going to take a break now. There's a poster session outside, and we're going to start back at 2:40. Thank you, Cori.